Hi everybody, this is my third attempt at recording this mini lecture due to various technical difficulties, so let's hope the third time's a charm. Welcome to this mini lecture on population genetics. This lecture will focus on the Hardy-Weinberg principle and how to use the Hardy-Weinberg equation. So let's get right into it. You might remember Darwin and his study of finches. Here he is seen below. So Darwin and his finches have largely shaped how we think about natural selection and evolution. In many ways, I guess, Darwin was the first and maybe the only famous population geneticist, even though that term wasn't even in use at the time. More recently, geneticists have moved beyond these simplistic comparisons of physical traits like Darwin and his finch beaks to make claims about the pressure of natural selection on traits and begin to use modern tools like DNA analysis, microscopic imaging, and enzymatic assays to look at the genetic changes that occur due to natural selection and how this affects the evolution of a population over time. Today, we will learn more about population genetics using the work of Dr. Colette St. Mary from the University of Florida for context. Dr. St. Mary, this is her here, studies traits that contribute to reproduction success. So we will use her work to set the context. She works with these banded coral shrimp over here, really cute things. This is a saltwater shrimp. So we'll use her work with banded coral shrimp to dig deeper into this week's topic of population genetics and the Hardy-Weinberg principle. But before we go any further, let's get a working understanding of what population genetics means. The field of population genetics uses the genetic composition of biological populations and changes in that genetic composition over time to show how genes and traits influence natural selection. Population geneticists use mathematical models to provide evidence of this work. It really all boils down to something we talked about a lot in Module 2, alleles. We will be using allele frequency this week. Allele frequency here tells us how often an allele appears in a population. Allele frequency is dependent on a number of environmental factors, but what it really comes down to is this. Organisms with traits that improve survival live on to pass those traits down to their offspring. Organisms that have traits that make survival difficult tend to die out. Therefore, survival supporting traits are naturally selected to stay in the population, and survival unsupportive traits are naturally selected to leave the population. Let's take another look at this adorable shrimp. Dr. St. Mary has studied 5 genetic traits of this shrimp and their effect on survival. So here they are here. These traits are size of adult shrimps, molt intervals, or the time between moltings. And moltings, in case you weren't sure, are periodic shedding and regrowth of the shrimp's exoskeleton. So that happens as the shrimp grows. Settling, or the ability of an adult shrimp to find a good home and stay there. Growth, and finally fecundity, or the ability to produce offspring. So how good the shrimp, how viable the egg and sperm are for these shrimp. By studying these traits over several generations of shrimp, Dr. St. Mary and her team were able to identify relationships between these traits, which sets up research questions for future studies. Remember last week when we hinted at linked traits? This is what Dr. St. Mary's team is currently exploring. Is there linkage between the traits that improve survival success? So let's move on to the modeling part and see how population geneticists like Dr. St. Mary can use this information to develop models that predict allele frequency. By now, you've read through the section of the reading about Hardy-Weinberg, and this little walkthrough will help you practice using the Hardy-Weinberg principle and its associated equations to set you up well for your graded assessment on population genetics in this module. The Hardy-Weinberg principle states that a population's allele frequencies are basically stable until some evolutionary force acts on that population. What does this mean? If we think about Dr. St. Mary's banded shrimp, say that rising sea temperatures force these animals to move more often, this would disrupt the normal settling time. The rising sea temperatures in this case would be the evolutionary force that selects for animals who are capable of packing up and moving around more often. Over time, this trait would be selected for. Now, this assumes, of course, that there are no mutations, there are no migrations or immigrations that would cause mix of populations. There's no selective pressure, like a predator that prefers larger shrimp to smaller shrimp. It also assumes that there's an unlimited number of shrimp and that the shrimp will reproduce for infinity. Of course, this is all very unrealistic for the real world. So, I mean, we know mutations happen spontaneously and selective pressure is like a real thing. So this is why we can only use Hardy-Weinberg to predict a model and to check in with data with known data, not to give us exact real-time data. So here's the equation, the first equation for Hardy-Weinberg. P plus Q equals 1, where P represents the frequency of allele number 1, usually the dominant allele, and Q represents the frequency of allele number 2, usually the recessive allele. Now we remember from Module 2 that if there are only 2 alleles and each allele has a 50% chance to be expressed, then the possible alleles in P plus Q at 50% each will always equal 100% or 1. But we know that alleles don't really exist as single entities. We also know that in Module 2, when we talked about probability of alleles, we were really talking about the probability that a specific genotype would occur in one individual offspring. So we'll put together what we learned in Module 2 about alleles and probability with what we are learning now about frequency to understand how to use Hardy-Weinberg to make a prediction about how alleles present themselves not in an individual offspring, but in an entire population of the same species. Okay, so let's look at the equation more closely. We'll just pick it apart. So our equation is P plus Q equals 1. If we stick with the context of the banded coral shrimp, let's say the allele P represents the genotype for large shrimp, and Q represents the frequency of genotypes that make small shrimp. So in this case, PP would be the genotype for large shrimp. PQ is heterozygous, large is dominant, so PQ would make large shrimp, and then QQ would represent the frequency of genotypes that make small shrimp. All right, so let's go back to the equation. P plus Q equals 1. So this is, on this slide, is really just informational in case you ever wanted to know. There's 2 equations. One is P plus Q equals 1, and then one is more complicated, and what's the relationship? So I've made this slide to kind of explain that. So each offspring requires 2 parents in the organisms that we talk about in this class. So so each parent has expresses frequencies of alleles. So P plus Q equals 1 for one parent, and P plus Q equals 1 for 2 parents. As we've learned through meiosis and mitosis, these parents combine, right? When the gametes meet together, the sperm and egg, they combine their DNA, so we can multiply these two. P plus Q times P plus Q. The frequency the probability and frequency of the gene pool will still remain 100%. So now let's do some algebra. Do you guys remember how to multiply P plus Q times P plus Q? So first thing we'll do is we'll multiply P times P to get P squared, and then P times Q, which gives us PQ. And then we'll do the other side of the equation, Q times P, which gives us another PQ, and Q times Q, which gives us another Q squared. So put that, simplify that, P squared plus 2P, right? PQ plus PQ. Oh, that's what's missing. That's a Q. 2PQ. Okay, so PQ plus PQ is 2PQ plus Q squared equals 1. This is the second Hardy-Weinberg equation. Okay, so let's keep working with it. All right, so we had our two alleles in the population, one that gave the trait for large shrimp and one that gave the trait for small shrimp. We know that there are three genotypes that will exist: homozygous dominant, which makes large shrimp; homozygous recessive, which makes small shrimp; and heterozygous, which would still express the dominant trait of large shrimp. So in the Hardy-Weinberg equation, the second equation, the frequency that will get homozygous dominant, or P squared, the frequency that will get heterozygous genotype is 2PQ, and the frequency that will get homozygous recessive would be Q squared. So we can see that the evolution of the first simple equation, P plus Q equals 1, to this one, P squared plus 2PQ plus Q squared equals 1. Okay, don't freak out. Stay with me because we're going to do an example together to help this make sense. All right, so pause the video here and read these two paragraphs that set us up for our example, focusing on albinism. And then when you finish reading, when you get to the bottom, push play, and then we'll continue together. All right, so we know that according to what we have here, the frequency for albinism in North America is 1 out of 20,000, every 20,000 people, or 0.00005. So since we know that albinism is recessive, which it told us up here, we're going to assign it the letter Q, which is the allele for recessive frequencies. We are assuming that recessive traits are less prevalent, and that way will be a smaller number than the dominant trait, which is P. All right, so let's keep going. By taking the square root of both sides, okay, so let me just step back a little bit. All right, so we have our frequency of the recessive allele, Q squared, as 0.00005. We want to know what is the frequency of just Q. Okay, so this is the frequency of homozygous recessive, but what about the frequency of just one recessive allele? So taking the square root of both sides of this equation, we get Q equals 0.007. So the square root of 0.00005 is 0.007. That's just one Q, not Q squared. This means the frequency of the recessive albinism allele, so just the letter A, is 0.007, or about 1 in 140 people. So now that we know the value for one of the two variables, Q, in the Hardy-Weinberg equation, it's easy for us to solve for the other one, P. So first we have our, just as a reminder, we have our first Hardy-Weinberg equation, P plus Q equals 1. We can rearrange this equation. We know Q and we need to find P. So let's get P on the other side of the equation. By subtracting Q from both sides, we get P equals 1 minus Q. So now we can just fill in our value for Q, which is 0.007, and solve for P. So 1 minus 007 is 0.993. So the frequency for the dominant allele, the one that does not confer albinism in the population, is 0.993, or about 99 out of 100 people. Okay. Now that we know the individual frequency for the dominant P allele and the recessive alleles, we can use the second Hardy-Weinberg equation to tell us the frequency for all three of the genotypes possible: dominant, recessive, and heterozygous. So let's go back to the equation for the three genotypes, P squared plus 2PQ plus Q squared equals 1. We can easily plug in what we already know using our own values of P, 0.993, and Q, 0.007. Right, so P squared would be 0.993 squared plus 2 times PQ, times 0.993 times 0.007 plus 0.007 squared, right? Q squared. All right, so when we do this calculation, what do we get? We get that P squared, or the predicted frequency of homozygous dominant individuals, is 0.986, or 98.6% of the population. We also see that the predicted frequency of heterozygous individuals, our 2PQ, is 0.014, or 1.4%. And Q squared, or our predicted frequency of homozygous recessive individuals, is 0.00005, or 0.005%. All right, so people who are carriers for the trait, that's these heterozygous individuals, are far more common than most people imagine, right? 1.4% of the population. And there are roughly 278 times more carriers than albinos, but clearly the vast majority of humans is 98.6% that are heterozygous dominant and do not have any of the alleles for albinism. All right, so now use this exercise, rewind if you need to and go back through it, and let's see if you can do an example on your own. Okay, so pause the video here and take a moment to read this screen, these 3 paragraphs plus 1 sentence about sickle cell disease, or SCD. Okay, so once you've read this, just write down on your paper, make a note that for African Americans, the frequency of homozygous recessive, or Q squared, is 1 out of 365 people, or 0.003, and the frequency of homozygous recessive for Hispanic Americans is 1 out of 16,300 people, or 0.00006. Okay. Now, you're going to do 4 things. First, you're going to start by writing out the 3 genotypes for sickle cell. Homozygous dominant, people who do not have sickle cell disease. Homozygous recessive, those that have sickle cell disease. And heterozygous, so those are people who carry the sickle cell trait but do not have the disease. Then, please decide if you want to calculate just one, the frequency for African American or the frequency for Hispanic Americans, or if you want extra practice or you're enjoying it, you can just choose to do both. Then you'll use the Hardy-Weinberg equations to find the frequency of the 3 genotypes in the US, and then take those answers and check them by putting them in the Google form on the main assignment sheet. If you get it wrong, you can try again, but also there is a solution, the solution to both frequencies for African Americans and Hispanic Americans. You can access them on the main assignment sheet. Okay, so give that a try, and we're going to use this information that we learn here in our live study session in our quote-unquote lab for this week. So hope to see you at live study session. Check me out at office hours if you have any questions about this material. Send me an email, and good luck.